Hello aspirants, welcome back to our channel Food Tech Online. Hope you all are doing well. So today we will be covering MCQs based on entrance exam for ICAR PG. So let's proceed. So the first question is, the first phase of a growth curve is, options are option A log phase, option B lag phase, option C gamma phase, option D both A and B, and the correct option is option B, lag phase. So this question is all about the bacterial growth curve where there are four stages. The initial stage is called as lag phase, then is the log phase or the exponential phase, then is the stationary phase, and lastly the decline phase. The first phase is called as lag phase where the bacteria or any organism takes some time to acclimatize to the surrounding medium. Resistance is mediated by, options are option A episome, option B plasmid, option C cold plasmid, option D both B and C, and the correct option is option B plasmid. The bacteria that resist the treatment with more than one antibiotics are called as multiple drug resistant organisms or the bacteria. These are mainly found in long-term care facilities or the hospitals, and they are very resistant to the multiple drugs. So herein there are two terms, namely the episome and the plasmid. Both are the lengths of DNA existing either in the cytoplasm or attached to chromosome of the bacterium. The only difference between them is plasmids do not integrate into genome and can be seen in the prokaryotes, while the episome is seen in the eukaryotes and it will integrate. Thus, the plasmid is the correct option. Question number 3. In gram-positive and gram-negative bacteria, the electron transport contains, options are option A naphthoquinone, option B plastoquinone, option C ubiquinone, and option D both A and B. And the correct answer is option A naphthoquinone. Question number 4. Growth in a closed system affected by waste product accumulation and nutrient limitation is called as, options are option A batch culturing, option B escus, option C fruiting body, option D sporangiophore. So the correct answer is option A batch culturing. So there are three methods or three ways in which microbes can be grown. It can be the batch process, the fed batch process, or the continuous process. So specifically in the batch culturing process, the nutrient medium is initially sterilized and then inoculated with microorganisms which are allowed to grow in a controlled atmosphere of gaseous environment and temperature. Nothing much is added except oxygen if there are aerobic microorganisms and antifoaming agents, acid or base to control the pH. Initially in the lag phase, microbes are adjusting to the environment, followed by the growth or the exponential phase where the primary metabolites are produced. In the stationary phase, the secondary metabolites are produced. Once the medium or the nutrients are taken up by the microorganisms, there is nutrient limitation and thus microbes produce waste. So there is waste product accumulation and this process is called as batch culturing. Question number 5. Cells are active and synthesizing new protoplasm. This stage of growth is called as, options are option A lag phase, option B stationary phase, option C log phase, and option D all of this. And the correct answer is option A lag phase. In the bacterial growth curve, as there are four stages, that is the lag phase, log phase, stationary, and the decline phase, the very initial phase is called as lag phase. In this, the medium is initially filled with nutrients, and as the cells grow, they take up nutrients and convert into new materials. So in the very first stage, as the nutrients are fresh and are initially added, these are required for the synthesis of the protoplasm and they act as a source of energy. Thus, the cells are very active and they synthesize new protoplasm during the very first stage of lag phase. Question number 6. Which of the following tissues can metabolize glucose, fatty acids, and ketone bodies for ATP production? Here, the ATP refers to the adenosine triphosphate production. The options given are liver, muscle, brain, and RBC, that is red blood cells. And the correct answer is option B, muscle. So, in our body, we have the glucose, which provides energy, the fatty acids, uh, which provide energy right after the carbohydrate source, that is glucose, and lastly, the ketone bodies, which are essential for the ATP production. So, in the liver, the ketone and the fatty acids are synthesized, and these are consumed and then converted into energy. The brain utilizes glucose as well as fatty acids, and RBCs don't consume any of the glucose or the fatty acids or ketone bodies. Here, the correct option will be muscle because the skeletal muscle utilizes all three, that is the glucose, fatty acids, and ketone bodies, and it is taken up for the ATP production. Question number 7. Rapid bacterial growth phase is known as. Options are option A, log phase, option B, lag phase, option C, lag phase, option D, none of this. And the correct answer is option A, log phase. So in the bacterial growth curve, as I mentioned earlier, there are 4 stages. Initial phase is called as lag phase. The second phase is called as log or the exponential phase. The third is the stationary phase, and the last is the decline phase. So initially the microbes are adjusting to the medium, and once they are adjusted, they start growing rapidly in the next phase, which is also called as log or the exponential phase. The primary metabolites are synthesized in this log phase, and thus there is a rapid bacterial growth. Question number 8. Clostridium welchii spore formation can be induced only on a specified medium such as. Options are option A, Wilson-Blair medium, option B, McConkie medium, option C, Elner medium, and option D, Thai-Martian medium. And the correct answer is option C, Elner medium. So this Clostridium is one of the species where the medium has to be rich in protease, peptone, and yeast extract. So the Elner medium here becomes the right answer as it is rich in the protease, peptone, yeast extract, and also the carbohydrate source, which is essential for the spore formation. So the strains of Clostridium, such the specifics such as the Welchii, vary in their ability to form spores in glycerol and inulin broth. Thus, Elner medium is the right answer. Question number 9. Mycotoxins are formed during the end of which phase? So options are option A, lag phase, option B, log phase, option C, death phase, option D, stationary phase. And the correct answer is option D, stationary phase. So in the bacterial growth curve, there are 4 stages. First is the lag phase, second is the log or the exponential phase, then comes the stationary phase, and the last one is the decline or the death phase. So the primary metabolites are normally synthesized during the exponential phase, and during the end of the stationary phase, the secondary metabolites are produced. Now the secondary metabolites are synthesized in response to the lack of nutrients or the environmental stress, as all the nutrients are getting depleted towards the end of the cycle. And the mycotoxin is also an secondary metabolite. Thus, it is formed during the end of the stationary phase. It can be very toxic to the humans as well as animals, and it can also cause diseases and death in both humans and animals. Question number 10. Bacteria which need oxygen for growth are called. Options are option A, thermophilic bacteria, option B, microaerophilic bacteria, option C, facultative anaerobic bacteria, option D, mycobacterium. And the correct answer is option B, microaerophilic bacteria. Depending upon the gaseous environment, if needed, the bacteria can be grouped into 2 categories. One is the aerobic bacteria that require oxygen for their growth, and second category is anaerobic bacteria which doesn't require oxygen for their growth. But there is one more class called as facultative anaerobes, which are aerobic in nature but can still survive in absence or lower concentration of oxygen. The thermophilic bacteria are not gaseous dependent but temperature dependent, where the thermophilic bacteria can grow with temperature range above 40 degrees Celsius. Question number 11. pH required for the growth of bacteria is, options are option A 6.8 to 7.2, option B 5.6 to 8.2, option C 3.0 or 3 to 6, option D 8 to 14. And the correct answer is option A 6.8 to 7.2. Now the bacteria are generally neutrophiles, that is they best grow at a pH range closer to 7, that is the neutral pH range. Acidophiles are the bacteria which grow at a pH near to 3, and the alkaliphiles are the organism that optimally grow between the pH of 8 to 10.5. Extreme acidophiles and alkaliphiles grow slowly or not at all near the neutral pH. Thus, the pH required for the growth of bacteria is the neutral pH range, which is 6.8 to 7.2. Question number 12. The ion that is required in trace amounts for the growth of bacteria is, options are option A calcium, option B magnesium, option C cobalt, option D sodium. And the correct answer is option C cobalt. Bacteria require a supply of inorganic salts. So there can be the requirement of the anions like the phosphate and sulfate anions, or it can be the cations like the sodium, potassium, magnesium, iron or calcium. But there is another ion called as cobalt, which may be required only in trace amount for the betterment of the bacterial growth. Question number 13. The reproduction rate is equal to the death rate in which stage of the bacterial growth curve? And the options are option A decline phase, option B stationary phase, option C lag phase, and option D log phase. And the correct answer is option B stationary phase. As we know, the bacterial curve has four phases: the log phase, the lag phase, the stationary phase, and the decline phase. In the lag phase is the initial period where the bacteria are adjusting to the environment, followed by the exponential phase where the population grows in double, followed by the stationary phase where the bacteria consume the available nutrients and produce the waste. When the nutrient supply is depleted, the growth rate enters a stationary phase and the number of viable bacteria remains the same. During the stationary phase, the rate of bacterial cell growth is equal to the rate of the bacterial cell death, and thus when the cell death becomes greater than the growth rate, then the population enters the last phase, which is the decline phase. Question number 14. The principle in microbiological assay is, option A, at certain range the concentration of growth factor will bear a linear relationship to the amount of nutrients added. Option B, concentration of growth factor have a linear relationship with the growth of the organism. Option C, both A and B, and option D, none of the above. And the correct answer is option B, concentration of growth factor have a linear relationship with the growth of the organism. Question number 15. If the source of energy for bacteria is from chemical compounds, they are said to be, options are option A phototropes, option B autotrophs, option C chemotropes, and option D chemolithotropes. And the correct option is option C chemotropes. Now the bacteria, depending upon the source of energy requirement and their ability to synthesize essential metabolites, they are classified. So the phototrophs are the bacteria which derive energy from the sunlight, whereas the chemotrophs are bacteria which obtain energy from chemical reactions. Then there are autotrophs, which where the bacteria can synthesize all the organic compounds, and the chemolithotrophs are the organism that can obtain energy by synthesizing inorganic compounds. Question number 16. Sulfur can be utilized by bacteria in the form of, Options are option A organic compounds, option B inorganic compounds, option C elemental compound, and option D all of the above. And the correct answer is option A organic compounds. Now sulfur is normally occurs in the form of the organic form, the inorganic form, and the elemental compounds. Now, sulfur is required uh, to synthesize the sulfur containing amino acids and certain type of vitamins. So, depending upon the organism, the sulfates, the hydrogen sulfide, or the sulfur containing amino acid may be used as a source of sulfur. So normally the bacteria can synthesize only the organic compounds, and there is one more organism called as sulfur reducing bacteria, which is specifically used to synthesize sulfur from the inorganic compounds. Otherwise normally the bacteria can synthesize only the organic form of the sulfur. Question number 17. The vitamin required for lactobacillus species is options are option A riboflavin, option B niacin, option C pyridoxine, and option D folic acid. And the correct answer is option B niacin. Now these are all the B vitamins, and the B vitamin is the water soluble compounds. Riboflavin is vitamin B2, the niacin is vitamin B3, the pyridoxine is vitamin B6, and the folic acid is vitamin B9. Now, the bacteria like the lactobacillus species require niacin for the growth because the cell wall is made up of peptidoglycan layer. So, niacin is important in making the cell walls of the bacteria. Question number 18. Vitamin K is produced by species. Options are option A Lactobacillus species, option B Bacillus anthracis, option C Bacteroides, Meningospecticum, option D all of this. And the correct answer is option A Lactobacillus species. Vitamin K is fat soluble vitamin that exists in the forms of vitamin K1, also known as phylloquinone, and vitamin K2, also known as menaquinone. It has very important function in our body, that it helps in blood clotting and thus in the reducing risk of the heart diseases. It is normally synthesized chemically, or it can be also produced in the nature and by microbial fermentation. In the nature it exists, especially the vitamin K1, uh, for example in green vegetables, and vitamin K2 in meat, eggs, or animal products, or it can be also synthesized by the bacteria. So there are some species which synthesize this, and the major species are known as the bacillus species or the lactobacillus species. Mainly they break down the dairy products, especially the cheese, and it can be produced. There is one more bacterium called as Flavobacterium meningosepticum that also produces vitamin K. Question number 19. The bacteria which can grow at 0 degrees Celsius but which grow at 20 degrees Celsius to 30 degrees Celsius are known as, options are A psychrotrophs, option B facultative psychrophiles, option C average psychrophiles, option D mesophile. And the correct answer is option A psychrotrophs. Psychrotrophs are the organism which are able to grow at 0 degrees Celsius, but their best temperature range is 20 degrees Celsius to 30 degrees Celsius. And these organisms cannot grow at temperature above 40 degrees Celsius. They are normally encountered in low temperature food spoilage because they can operate or grow at refrigeration temperatures. Mesophiles is another category where the organisms grow best at temperature range around 20 to 45 degrees Celsius. Psychrophiles are of two types, and they normally operate at temperature range around 0 to 40 degrees Celsius. They are having two types. One is the facultative psychrophiles and the second one is the obligate psychrophiles. Now the obligate psychrophiles can grow at temperature not more than 30 degrees Celsius, whereas the facultative psychrophiles can grow up to temperature 40 degrees Celsius. Question number 20. The orderly increase in the quantity of the cellular components is known as. Options are option A reproduction, option B growth, option C binary fission, and option D none of this. And the correct answer is option B growth. So the bacteria reproduce mainly by a process called as binary fission. It is a form of asexual reproduction in which organism is divided into two parts, each carrying one copy of the genetic material. Once the cell is divided, it starts to further multiply or replicate by increasing the cellular component within it. And this orderly increase of the cellular component is called as growth of the microorganism. Question number 21. The most common mode of cell division in bacteria is. Options are option A binary fission, option B transverse binary fission, option C longitudinal binary fission, option D none of this. And the correct answer is option A binary fission. Uh, the bacteria or any of the organism normally divide by asexual reproduction, that is called as binary fission, where it divides into two equal parts and both the parts carry the same copy of the genome. So there are three types of binary fission. One is the transverse binary fission, second is the longitudinal binary fission. But the most common cell division which occurs in overall all the bacteria is the binary fission. Question number 22. How much time a bacterium take for the complete duplication? Options are option A 30 minutes, option B 10 minutes, option C 20 minutes, option D 25 minutes. And the correct answer is option C 20 minutes. Most of the bacteria grow faster and they normally divide using binary fission where they splits into two parts. Thus the bacterial population double in size and the most or the fast growing bacteria can take up to 20 minutes for its growth or its complete duplication. Fast growing bacteria like E. coli take only 20 minutes. Question number 23. The generation time is. Options are option A time it takes for a population of bacteria to double in number. Option B the total division of the cell during its lifetime. Option C the total number of cells formed, and option D none of these. And the correct answer is option A the time it takes for a population of bacteria to double in number. The generation time is also called as doubling time because a cell divides using binary fission and it divides into two cells by copying all the genetic material exactly into half. Question number 24. In bacteria, the increase in population is in the manner. Options are option A geometric progression, option B multiplication, option C doubling, and option D none of this. And the correct answer is option A geometric progression. Bacteria generally replicate using asexual reproduction, that is called as binary fission. The cells split into two parts and thus increasing in the number of the cells by geometric progression. That is, one cell will divide into two, then the two cells will divide into four, then further it will divide into 8. So this is a geometric progression and not multiplication or doubling. Question number 25. During exponential phase, growth rate is. Options are option A same as the generation time, option B is reciprocal of the generation time, option C time required for the population to double, and option D rate of doubling population. And the correct answer is option B reciprocal of the generation time. During the exponential growth, the growth rate, that is the number of generations per hour, is the reciprocal of the generation time. Growth rate is the slope of the straight line obtained when the log number of cells is plotted against time. With this, we have completed all 25 questions. So, thank you all. One more request to you all. Please install our F-FoodTech online and the link is already given in the description box. This will help you to prepare for any of the entrance exam related to food technology PG course. Thank you.